Welcome to The Explainer. Today we're diving into something absolutely mind-bending regarding the future of biological design. Think about the stark contrast between two very different worlds. On one hand, you have the messy, squishy, chaotic, and highly unpredictable world of biology, and on the other hand, you've got the rigid, flawless, zeros and ones world of software engineering. For decades, these two domains have operated in completely separate universes. But what if we could merge them? What if we could take the strict, predictable principles of computer engineering and apply them directly to the incredibly complex building blocks of life itself? Just think about that for a second. What if you could compile and program a living biological organ the exact same way you program a silicon microchip? The stakes here are absolutely massive. Right now, there are over 100,000 people in the United States alone waiting for a life-saving organ transplant. What if, instead of waiting for a donor, an engineer could just open a software program, write a few lines of code, and literally compile a living, breathing, functional kidney? That is the grand challenge we are unpacking today. We're looking at a theoretical framework where researchers can specify, compile, and verify biological designs, scaling all the way from basic genetic modules right up to full, living organisms.
Here is our roadmap for today. We'll start with programming lifelike software, move to the fragmented biology toolkit, explore the silicon-to-cell analogy, break down the Bio OpenLane pipeline, look at the roadmap to synthetic organs, and finally touch on safety, ethics, and governance.
All right, part one, programming lifelike software.
So, Bio-HDL stands for Biological Hardware Description Language. In the computer world, a hardware description language is the foundational code used to architect and design computer chips, while Bio-HDL is essentially the source code for biology. Picture what this actually looks like for a scientist. You aren't standing at a lab bench with a pipette, manually mixing chemicals in a beaker and just hoping for a specific reaction. Instead, you're sitting at a keyboard. You're typing out the exact behavioral specifications for a biological system. You're compiling it, and you're verifying that it works in a virtual environment before it ever exists in the physical world. It's a proposed platform that completely mirrors electronic design automation, but for living things.
Moving on to part two, the fragmented biology toolkit.
Biological systems are incredibly complex because they span so many different scales. We're talking about the molecular level, genes, single cells, tissues, organs, and finally entire organisms. And the crazy thing is, we actually have brilliant software tools for these individual scales right now. Take AlphaFold, for example, which recently revolutionized our entire understanding of protein folding at the molecular level. It's a monumental achievement. We have Rosetta for molecular modeling, SBML for defining cellular pathways, and SBOL for standardizing genetic designs. We even have tools like PhysioCell to run beautiful 3D simulations of tissue growth and Neuron for mapping complex neural systems. But there's a massive underlying problem. These incredible pieces of software are trapped in their own silos. They're highly specialized, brilliant fragments of a toolkit that are completely disconnected from one another.
Simply put, no single language spans all biological scales. We're missing that unified end-to-end flow. You can't just take a high-level tissue simulation of a beating heart in PhysioCell and automatically compile it down into the specific genetic parts needed in SBO. That bridge to cross biological scales just doesn't exist yet. And you know, biology is uniquely unforgiving here. A tiny microscopic tweak in a single gene can create this massive butterfly effect that completely ruins the 3D structure of an entire organ. Without a unified language to translate between those scales, designing complex biological systems remains incredibly slow and error-prone.
Which brings us to part three, the silicon-to-cell analogy.
To build that unified language, the industry is looking at how computer engineers solved this exact same problem decades ago with electronic design automation. Let's break down this analogy. It's actually really cool. When designing a computer chip, engineers use a standard cell library. This is a collection of pre-tested, reusable electronic components. An engineer knows for sure that a specific resistor will always behave the exact same way. The biological equivalent to this is a library of biobricks or iGEM genetic parts. Think of these as the literal Lego bricks of DNA. They're standardized, reusable biological parts, like promoters, genes, and genetic switches, that we can confidently snap together to build complex genetic circuits, knowing exactly how they'll perform.
Now, this next part of the analogy is where the stakes get incredibly high. In chip design, engineers run a design rule check to make sure the wiring won't short-circuit. If there's a bug in your web browser, it crashes, you get the blue screen of death, and you just restart your computer. No big deal. But in biology, a bug is a lot more serious. A bug means the cell dies from metabolic overload. It means toxic buildup. Or worse, a bug means horizontal gene transfer, where a cell uncontrollably mutates and spreads. So in our Bio HDL system, the design rule check becomes a strict biological feasibility check. Before a single strand of DNA is ever printed, the software runs rigorous rules to debug the life form, preventing you from accidentally creating a lethal or biologically impossible gene network.
Finally, we have the manufacturing phase. A silicon chip goes to a massive, highly automated fabrication plant to be printed on wafers. In our biological workflow, the compiled design goes to a roboticized biofoundry. Now, these are not your traditional college science labs. Imagine a facility filled with the low hum of machinery, where precise robotic arms move microscopic, nanoliter droplets of liquid with absolute perfection. These automated labs take the digital code, the ACs, Ts, and Gs, physically assemble the DNA constructs, execute the cell culture protocols, and actually manufacture the living, breathing cells at scale.
Part 4. The Bio OpenLane pipeline.
Okay, so we've got the genetic Lego bricks. We've got the automated robotic labs. We have the individual simulation tools. But how do we actually string them all together into one seamless, automated workflow without the whole shebang just falling apart?
The proposed architecture to solve this is called Bio OpenLane, and it operates as a 5-step, closed-loop journey. Let's walk through exactly how an idea becomes a living organism. Step 1 is specification. This is where human intent enters the system. An engineer uses Bio HDL to write a high-level command. And they aren't writing out raw DNA, they're writing logic, something like build a vascular network designed to filter toxins at this specific rate. Step 2 is synthesis. This is where the magic happens. Specialized tools, like a program called Cello, act as a biological compiler. Cello takes that high-level human logic and translates it, literally spitting out the exact sequence of adenine, cytosine, thymine, and guanine, the DNA base pairs, needed to make that logic a reality. Step 3 is layout. Now that we have the DNA sequence, we need to know where it physically goes. This step defines the 3D spatial arrangement of the tissue layers, determining exactly how the cells will be organized structurally. Step 4 is fabrication. The digital blueprint gets sent over the network to the wet lab biofoundry we just talked about, where robotic arms assemble the DNA and grow the actual tissue. And finally, step 5 is verification. The system compares the physical lab results against the original computer simulation to ensure the living tissue performs exactly as designed. It's a complete automated loop from an abstract thought to a biological reality.
Let's look at Part 5, Roadmap to Synthetic Organs.
Taking this from a theoretical 5-step pipeline to a daily reality is a massive, multi-decade undertaking, but the foundation phase is practically tomorrow. Between 2026 and 2028, researchers aim to lay the Bio HDL foundations, finalizing the syntax and standardizing those biological parts. By the late 2020s and early 2030s, we move to single-cell prototyping, perfectly automating simple microbial gene circuits. From 2030 to 2035, the capability jumps significantly. The language will extend to spatial designs, allowing us to compile simple, 3D-printed tissues and basic organoids. And then we hit the true sci-fi milestone. By 2035 to 2045, we are looking at full organ and organism level integration. We're talking about designing an entire, complex, functional organ, like a heart, completely from scratch using Bio HDL. And beyond 2040, we enter an era of advanced machine learning optimization, where AI refines these biological designs way faster than humanly possible.
And finally, Part 6, Safety, Ethics, and Governance.
Look, engineering life carries a profound responsibility, and you absolutely cannot build a system with this much power without some serious, baked-in safeguards. Biology is deeply analog, and unlike silicon, living things evolve, they mutate, and they adapt. A buggy mobile app is annoying. A buggy synthetic organism released into the environment is a potential ecological disaster. Because of that, the framework heavily stresses the need for anticipatory biosafety. A cornerstone of this is digital twin testing. This means we rigorously and exhaustively simulate the organism in a virtual environment, pushing it to its absolute limits before a single drop of liquid is ever mixed in the wet lab. Furthermore, this requires unprecedented global governance. The industry is proposing strict international standards, much like the JEDEC standards that govern the global semiconductor industry today, but uniquely applied to living, breathing designs to ensure biological security worldwide.
The ultimate goal is a future where we can compile biological designs into living systems as predictably as we compile software into silicon. It's a vision that requires unifying decades of bioscience, software engineering, and global governance frameworks. If it works, it fundamentally rewrites humanity's relationship with the building blocks of life. So I want to leave you with this. If we truly crack this code, and we gain the ability to compile biology exactly like we code apps, what will you choose to design first? And maybe more importantly, who gets to decide what we should or shouldn't print into existence? Thanks for joining me on this explainer, and keep questioning the incredible mechanics of the world around us.